Total coliforms, and specifically fecal coliforms, can be found in water supplies. A specific type of fecal coliform would be E. coli, and this is a bacteria that is normally found in the intestines of humans and animals. Sometimes E. coli can contaminate our water supplies. This can lead to medical issues such as diarrhea and dehydration. Since E. coli can have some medical issues, it is important to test our water supplies, to test water samples. What has been done by my professor, Joseph Frenzel, and myself is use and compare 3 methods for E. coli. The first method used to test for E. coli is the Quanti-Tray method. This method, which is new at Grace College, uses a machine called the Quanti-Tray Saylor 2X. The Quanti-Tray Saylor 2X seals trays in less than a minute. The trays that are sealed by the machine are filled with a 100 milliliter environmental sample from the Tippecanoe River by a waste facility, and the 100 milliliter sample is mixed with colort, which will be the indicator for coliform and E. coli. Once the trays are sealed, they are incubated for 24 hours to obtain results. For the second method, test tube method uses 9 milliliters of EC medium with mug in each test tube and 1 milliliter of environmental sample in each of the first 5 test tubes. And the first 5 test tubes are 1 milliliter of the first 5 test tubes is pipetted into the next 5 test tubes, and then the second 5 test tubes are 1 milliliter of that is pipetted into the third and final set of 5 test tubes. And once all the 15 test tubes are pipetted in, they're incubated for 24 hours, and then the results are obtained. For the third and final method, it's the agar plate method, and this uses 2 types of agar, a sterile membrane filter, and a culture plate, and a 10 milliliter of environmental sample. The 10 milliliters of environmental sample is poured into a vacuum filter filtration, and then it's vacuum filtered in through a sterile membrane filter, and then placed on a culture plate containing the 2 agar, and then this is incubated for 24 hours to get results. The first method, which is for the Quanti-Tray method, these are 2 images. The first image shows yellow, and this is after the incubation. The yellow indicates coliform. In the second image, the fluorescence indicates positive for E. coli. For method number 1 results, the most important part to look at is the MPN values of upstream and downstream. For the 2 upstreams, it's 76.7 and 82, and for the 2 downstream, it's 520 and 580. I averaged up the MPN values of the Quanti-Tray method, and as you can see, the downstream has a larger MPN value as average rather than the upstream. And there's no standard deviation shown on this graph, but there are differences in the values from between the 2 upstreams and the 2 downstreams. The second method, which is the test tube method, these 2 images show positive for both coliform and E. coli. In the first image, as you can see, there is growth on the bottom and a bubble right here in the inverted tube, which indicates gas. This test tube indicates that there's a positive for coliform. In the second image, it shows two test tubes. The first test tube is fluorescing, and this indicates a positive for E. coli, and the next test tube next to it is just a comparison. This doesn't have any environmental sample, it just contains the broth. So the results in method number 2, the important part again is to look at the MPN values. In the two upstreams, 32 and 24, in the two downstreams, it's 100 and 470. I averaged up the MPN values, and as you can see, downstream has a larger MPN value as an average than upstream. I did not show standard deviation, but there is much difference between the two upstreams and the two downstreams. The third and final method, these two images show, in the first image, it shows a positive for coliform. The gold specks on it show these are colonies on the agar plate. In the second photo, it shows fluorescent dots on the agar plate, and this indicates a positive for E. coli. The results in method 3, this is different. There's no MPN value, but it's calculated as the total colonies per 100 milliliters of water. The most important part of looking at this graph is, at this table, is the fluorescence total colonies per 100 milliliters of water. In the two upstreams, it's 240 and 200. This is the total colonies in that place, and the three downstreams is 210, 30, 570, and 170. I averaged the total colonies together, and as you can see, downstream is again has the most amount of E. coli present, and there is more of a standard deviation in downstream than upstream, because there's a 30 value, and then the one is 570, and they're just different values for it. And conclusion. Results show a high MPN values for downstream. So in all three methods, all of them indicate that downstream has the highest MPN value for how many E. coli is present in the water samples. And plus upstream also has high MPN values of E. coli because you could detect them, but even more so in downstream. And keep in mind, the water samples were taken in a river next to a water facility, both upstream and downstream. And then in the tube method, there was more variation. So the results in the test tube method, when compiling the data, shows that there was different amounts in them. The results show that all three methods can be used. So all of them gathered data for the E. coli testing. So all of them can be used for E. coli testing. And finally, Quanti-Tray did find results that correlate with the other two methods.